Okay, it's a great honor to introduce our work to you guys. Hopefully, after the talk, we can do more kind of a collaboration work together. And initially, I want to start to address the question why we need a better model. I don't think I need to argue too much because the current preclinical model not really doing a great job. What are the most commonly used models for industry and academic field? Actually, most people really just use a cell line, in vitro, in vivo. That's a common practice. But when you look at the cell line and how different and they show up from a real human tumor. And this picture, actually, I just gathered a corner of prostate cancer specimen. If you look at the higher magnification, you can notice that you have a benign tissue, benign mixed with cancer, carcinoma in situ, low-grade prostate cancer, and then gradually go all the way to the perineural invasive cancer. So even in one piece of real world, you contain multiple type of cells, benign cancer, even different type of cancer cell. But when we look at the cell line, and this is a typical PC3 cell, so those kind of cell, normally we use them claim as representative for prostate cancer. That's a very corner vision, actually, tunnel vision, in other words. Even when you put the cells inside in mice, we call xenograft. You can see LinCap xenograft, PC3 xenograft, DU145 xenograft. So they are very different from what you see from a patient. So the significant difference certainly will make, big difference in outcome. So one of the ideas is can patient-derived cancer tissue xenograft be a better solution for that? And of course, then outside the patient, what could be the most relevant model that may make a patient tumor? Probably the patient tumors themselves are the best. But the challenging was, and when we came into the picture a number of years ago, when we start to look at what happening in this kind of field, and we noticed that historically, prostate cancer xenograft has been extremely inefficient. So this is what we put in our very first paper. The paper reviewed 3 times by cancer research, but the review did not like the background. They want us to change it. But we stay with what it was. And basically, we're trying to say a lot of efforts has been put on, but it's so inefficient. It's not so successful. So that's why you need a better method to improve that. What we did is, that time, it seems that everybody in the field, once they get a patient tumor, they graft them subcutaneously in mice. That method is kind of very simple, but the drawback is the take rate is extremely low. The reason for that, actually, we're getting a better understanding why that's happening all the time. Because when you get a new piece of tissue, move to a new environment, all the angiogenesis cannot happen overnight. But before the new blood vessel establishing, the cancer cell or any cell inside the graft still need to get nutrition to get rid of the waste. And how they can get those process done. And in that case, actually, the blood fusion became a key issue. And in terms of blood supplying, the subcutaneous side is very bad. And you look at your skin is normally not red. It's white or black or yellow, whatever. And we were thinking that's probably the main reason. And the subcutaneous side is probably not a good place for doing graft, particular at the very first 24, 48 hours. So what we did is, if we get a piece of early stage prostate cancer or later stage, whatever, then we put them, actually we graft them to multiple grafting site in the animal. And here only demonstrate subcutaneous graft, also topic graft, sub-renal graft. I just want to do the comparison. And to cut the long story short, and no surprise to us, but a surprise to many people. So whenever we graft the human tissue and the renal capsule, the take rate basically is 100%. The reason we put 95%, we're leaving a little bit of room to show the modest attitude. Okay, so unless you have a technical problem, the survive is not an issue. All the graft can survive well, but not necessary to say all the graft can be rapid growing ones, because that depends on the nature of the graft tissue. So over the years, we consistently prove this kind of method really work. I just want to show you a few examples. Here, on this corner, you can see the lower power. This is a piece of prostate tissue, mainly benign, and this is a mouse kidney adjacent to the graft. If you just focus a corner here, and you can see this, all the image come from this tiny corner. This is a typical pain lesion, and you can see the basal cell is partially lost. But the cell, in terms of the proliferative rate, and only very few cells in cell cycle, which that reflect what you see in the patient. And then if you focus the two best differentiation marker, androgen receptor and the PSA, you can see how beautifully they maintain, the expression profile. So this is a piece of prostate cancer tissue. You can see this is a donor tissue. If you graft them into the animal in the castrated mouse, and you can see the cancer cells start to shrink. And this kind, if you give them a good condition, they look very similar to the donor tissue. So this suggests the tissue and the renal capsule also responds to androgen manipulation. And if you look at this one, this is another one. And this is a low-grade and prostate cancer. This come from control. If you treat with a docetaxel, this actually is a good responder. But if you look at the graft volume, you wouldn't get that concept. But if you look at the cellular level, you would see, okay, the cells and many of them are actually dead or dying. So, but even the cell dead, they may still occupy the space. But look at the cellular level, you still can learn a great deal from there. After that, and a lot of people, is wondering if the method is really working so well, how about the possibility to use the same method for other type of tumor? So that time we did not have Larry's support. We have to get whatever we get. So a lot of people came to us, whoever can bring the real collaboration, money, funding, whatsoever, we try those. And then this is an example, and come from a colon tissue. So before grafting, you see how they look like. And this is a graft and adjacent to renal kidney. And then you just look at high magnification. You see this how similar they are. And this is a piece of lung normal tissue. This is a kidney. And then this, look at the high magnification from this corner. And then when Jean LaRich saw this picture, and she was very pleased with the whole thing. And then she said, oh, I never really saw this kind of, happy, viable, normal lung tissue can exist in any kind of animal model system. So she was very pleased with that. And this is a kind of moderate differential lung cancer. I just give you a quick image before grafting and after grafting, and you can see the similarity. A lot of people working on this, and Jessie Cute is right here, June, and Margaret Sutcliffe, and also working for this project. And we also did a collaboration with Wang Lam, and his group checking the CGH profile. He found that before grafting and after grafting, the similarity is extremely high. The same methodology we use for ovarian cancer, and before grafting and after grafting, again we proved that that's pretty good. And we also using tissue microarray using multiple marker that's mainly used by ovarian cancer groups. And then we found that the similarity is extremely high. And then we published that paper. And also another question we asked, and some people challenging you, if you get a slow-growing tumor, you just graft them in an experimental condition, of course you wouldn't see significant change. And when you're comparing them with the donor tissue. And the question is, some tumor may have a rapid growing nature, which they are suitable for developing a transplantable tumor line. Once you're talking about those kind of models, are going to be really convenient because they're going to expand nicely. But the question is how far they have gone, and whether or not they still maintain similarity to their donor tissue. So this is an example come from ovarian cancer, and before grafting, those are the tumor line we call. Once we call them tumor line, they already passage in animal file for 5 generations. Generation is basically you put one set of a host, and then you harvest the tissue, regraft them to second host. That's called 1 generation. So in that case, any tumor line, if they can passage for more than 5 times, we start to call them tumor line. So for this, even after 5 generations, you still can see, the histopathology from that point of view, how similar it is. And also working with David Hausman's group to checking the genetic profile, we always find that the tumor line can always find their home. So we don't have to spend a whole lot of time to prove you how good the model is. And any model by definition is not true. So no perfect model, but this model probably the most close, most relevant model anyway. So this model have a lot of good properties. You can argue how bad they are, but if you argue that aspect, you can always have a lot of things come up. But this is the kind of model we are happy with. Yes, I think I make an argument we need a better model. This kind of model, it became practical. And the next step is really focused on how you can use the model to do application. I think application is the central piece of our efforts, because a model is a tool. Whatever you do, and you have to use the tool to address your clinical relevant questions. So I have to be, this slide makes me feel extremely happy. So this is the one actually over the past 5 years, 6 years or so, we developed more than 120 different tumor models. And what this number really tell you? This number really tell you 2 things. And probably this is the biggest number whatsoever in North America at least. So not to mention the quality is extremely high. So that's something within such short time, the lab has progressed really significantly. But we did not really voice that loud. And, but this is the data really tell you where we stand. So this is a list of ovarian cancer. You can see, just to give you kind of a ballpark ideas how colorful those kind of model look like. If you talk about ovarian cancer, there are many type of subtype of tumor, and even you talk about same type of tumor, and their biology, even they look like the same, but their biology very, very different. In this case, I just want to show you, and then you see the drug sensitivity to a standard chemotherapy, even they are the same kind of high-grade carcinoma. One is good responder, one is bad responder, one is partial responder. So it's not necessarily you're talking about one thing. One model cannot tell you the whole story. You need a panel. We will come back to that topic shortly. And this is the kind of a strategy we use. All the tumor line, we make them into tissue microarray. And then whenever you have any marker, any target, you can do a quick screening and to find a suitable model and whatsoever. So the idea is, how are you going to select a suitable model or maybe right model to use? Okay, you not only just pick up the model that support your hypothesis. If you really want to face with a clinical challenge, you have to challenge yourself in the right way. So what we're trying to do, and we're trying to put this all the information into a website, and then we call this one Living Tumor Center at a BC Cancer Agency. But Martin doesn't like the Living Tumor Center at a Prostate Center. So we're going to call the Living Tumor Unit at the Prostate Center. So that makes everybody happy. I take that point. But the central piece is kind of, and once you get into this website, let's say this is the meat of the whole thing. If you go to what kind of model you have, you can go to ovarian cancer. And then you click ovarian cancer, you will come up a whole list of ovarian cancer tumor line with a brief description of each tumor line. If you are interested in, let's say in this case, you are interested in BRCA1 germline mutant one, you click that, you will come up with a data sheet. The data sheet for each tumor line will have 2 or 3 pages. And then the data sheet will tell you and what kind of donor tissue look like, what kind of genetic or some case you have an expression profile. And then you come back the main thing, what's the tumor line look like. Then you may come up with idea, this is the kind of model, and I feel very comfortable for me to test let's say the PRP inhibitor for BRCA1 mutation ovarian tumor. So for this kind of things, actually, if you have a good hypothesis, what kind of the best model you use. And most people in the field using artificial, mouse cells and human cell, use and genetic engineering things. But in this kind of things, you will say this tumor carry those BRCA1 deletion. That's actually exactly the spontaneously developed from patient. So that could be maybe still is the only model in the world that carry this spontaneous, patient tumor with the particular deletion. So if you're interested in prostate cancer, you go to the prostate cancer field. Then you're going to have a whole list of our model. It's not a P63, not a DO145. This all come from patient, okay? If you're interested in, let's say, PCA1, and you are interested in metastatic one, then you go click there. You will come up with this one, and then say what is cytogenetic information, and if you graph them also topically, what kind of metastasis you see, and what kind of progression table you can look at. Actually, this is just a snapshot of the database. You will learn more over there. The data sheet will contain more and more information. the idea is trying to make the model more accessible and to everybody in the world in long run. So I just give you another example, and for anti-cancer development. A lot of people are interested in that, and for most of cancer, once you develop into, advanced cancer, so you do not have good cancer drug, that's for sure. So a lot of efforts put in this area. I don't have to educate you. And this is an example, and we did with Pfizer, so they want us to test a Sutent in ovarian cancer. So the whole idea is that if you get ovarian cancer and you put them in multiple group and test their Sutent to comparing with a standard chemo, and then in this case we even put them in combination with standard chemo. But the only thing is different is we not really just using one model. We tell them, hey, if you ask me to do the work, and I don't want to just get a corner view, I want to have a panel of model to test your drug. So they approve and they accept the concept. What's so interesting here? So so far we got a 5 model down. Initially we got a 4 model down. And the very first 3 models here, and you can see, just look at the control arm and Sutent, and the control arm and Sutent, and the control arm and Sutent. And you find Sutent has no effect at all, and in the model, while the standard chemo working pretty well, just based on the tumor volume, okay? And then at that time, everybody feel disappointed, and we never really find the targeted drug working better than chemo, and in all the model we test, and that kind of understandable. I come back to that point shortly. But in any case, that was a disappointment, and until we reach the number 4 model here. And we find the Sutent really work pretty well. And then the first question we want to ask, what's the difference between the 2 groups? Actually, at that time we find the first 3 are typical high-grade serous carcinoma. The number 4 is a clear cell carcinoma. So this is the image from the responder. This is a control. You got a very bloody tumor. And you see the Sutent one, actually they really anti-angiogenesis. It's a kind of a whitish. And so this kind of image, and by all means prove the drug really work. And so we got 1 paper almost ready for this one. But the lesson we learn from here is kind of fascinating. At the same time, and we went to the ovarian cancer meeting, and I joined the workshop, and then the clinical people gave their clinical trial data from phase 2, and we bring our animal data to them. Actually, I don't want to comment too much, and our data really gave them a lot of insight for them to go back to analyze their clinical phase 2 data. And another aspect was kind of interesting, because here, and Diane Miller look at this data. She's on this study, and from ethical point of view, she actually started put some Sutent on 2 of her clear cell patient, and it seems that 1 patient respond pretty well. And I don't know how the situation look like at this moment. But anyway, this is another example. I can give you a lot of examples, but I try to stay short. Okay. Another example is how to use the model for discovery. You cannot get a promotion without getting CIHR grant, NCIC grant. You have to prove you have a hypothesis, you have a good project. By all means, all the model developed, we don't have any penny that's specifically used for development model. Nobody support that. All the model and is kind of developed based on the founded grant, which you trying to say, we want to do this, and we want to use a model. Then we have to budget for some model development. So this is a very good example how we do with CITR and other funding agency. So people really like to go to the fancy part. And if you are interested in discovery in some way, and you have hypothesis driven and kind of at high level, and they seem to reveal intent to agree with you. So this kind of model can be used for discovery, kind of signature for good responder, non-good responder. In this case, I give you a different example. When I look at the whole thing, I was thinking one is early detection, one is treatment for metastasis, or prediction for metastasis. So those are the two major areas. If you ask me to go, that could be the area I like to go. So in terms of metastatic prostate cancer, you don't have good therapy. You don't have a good predictive marker to predict which early stage of prostate cancer could be more aggressive or less aggressive. So I think that can be really relevant. And then the question is, and this is something I learned, I like to share my personal point of view with you. Whenever you start a new project, you want to view that what's going on in the field, what's a normal common practice. If everybody does that, why they do it? If they are not so successful, why is that? If you want to jump into the field, you have to be sure you're going to be better than them. Then you have a chance to succeed. Otherwise, you're going to fight a losing battle all the time. So this is my personal experience here. So the same question I asked, I said, okay, a lot of big people and working in the field and trying to find such things. What's going on? What kind of a method they use? And forget about the mouse model, cell line model, or whatsoever. And once they care about a patient situation, what they normally do, they get a primary tumor comparing with secondary tumor. Okay, they want to find the differences between the two major populations. Sound very logical, right? Yes, and because all those geniuses do that. Okay, but to me, as a job act is quite clear. You see, as I point out earlier, you see how heterogeneous the primary tumor is. And actually the secondary tumor very heterogeneous as well. The higher variation you have, the higher N number you have. You need the big variation you're going to have. Actually, for this kind of big variation, nobody knows how many N number you need for any kind of analysis. Not to mention you have a powerful genetic approach whatsoever. Probably you're not going to have enough power to gain enough confidence. Even your mathematic power is enough, not necessarily your biological power is there. The second challenge is obvious. Because the primary tumor, the microenvironment is certainly different from the secondary one. That make huge impact because the microenvironment can affect your gene expression by 70–80%. What that really means even you have a metastatic associated gene sitting there, but that could be diluted greatly by your microenvironment effect. And the third challenge, how many people really have enough secondary tumor to study? I don't have. And Martin and Larry have some, but not too many, maybe 20, and certainly less than 50. And even you're consistent to collect some, the limitations are pretty much there. And in the whole world, and it's probably only 2 or 3 worm autopsy program can have luxury access to some secondary tumor, but most of us do not have it. So again, Those are the challenges. When you want to jump in, how you're going to use your kind of unique approach. Not only just unique, and kind of your hope is going to be better. That's going to leading to some fruitful kind of outcome. So this is our approach. And was a kind of a dream, kind of a number of years ago, but the dream actually came true. And so we know the prostate cancer is kind of heterogeneous. Our dream is, can we use our higher efficient xenograft system to dissect the heterogeneous population? The idea is that if you end up with one major population is metastatic and another population is non-metastatic, and then how wonderful it is. Like you get a twin brother from one parents, and then basically then this kind of comparison going to be really nice, and the noisy everything going to be small. Even you have artificial environment, but they subject to the same kind of microenvironment, right? Artificial treatment. So the comparison is still there. The background is the same. So the dream actually, as I said, came true. And this is the very first case. We got a piece of human prostate cancer tissue. We graft them through our kind of, this is a long story. I can come back to this one. In order to reach this two subpopulation, it took us 4 years to get it. But once we got there, it's so great. And this is the very first case. You can see the subpopulation, we call that PCA1. This is the tumor line, give you typical metastasis. And this one, the counterpart is a PCA2, we call them non-metastasis. And then you can check even from a cytogenetic point of view. Actually, you look at the chromosome number, they're actually diploid. They're not aneuploid. And do you need 100 chromosomes to make a cancer? The answer probably is not. And why you have 100 chromosomes? And probably, cancer also generated additional genetic alteration in due course. So this is the kind of situation you have to look at. That's why our tumor line in our hand, we try not to let them to see any petri dish. If we see that, we know that probably give you additional noisy, and that's going to complicate your study. So like anyone else, we based on just one pair. We did a 2-stage library analysis, and we, like anyone else. And a lot of people argue about that. The N number is not good enough. You need 100 of A, 100 of B. And if that's the case for comparing the primary tumor to secondary tumor, probably that 100 is still not good enough. But my argument is our sample is highly homogeneous, and that's one population against another one. So we just use a 2 library. We wish we could have more, but that time we don't. And we went on anyway. Just based on this 1-pair library, I have to tell you that we are very successful. We generated, we focus, we discover a few metastasis-associated target gene, and those target gene actually leading to 2 major CITR operating ground, and more to come actually. So this is one example, and Stephen is a student working in the lab, and we collaborate with Martin and Alan So in this case. So we did a comparison, and we identifying a gene called ASCP1 that time, just to give you a little bit of flavor, not necessary to say the ASCP1 is the one we want to go forever. I just want to give you a little bit of flavor to say how we take the candidate through all the validation. So that time the criteria for identifying a candidate gene is you want to have something never really reported in prostate cancer, but you don't like to go a highest risk candidate. You like to have some novelty, and that gives you certain degrees of safety, index there. And this is the one totally fulfill what we're trying to do. That time, never really report in prostate cancer, and locations are pretty hot, located on AQ24. That's the area everybody looking for something, and turn out this gene there. Then the first question we ask is this gene is a kind of a clinical relevant. If you find something, it's only interesting in your model, that's no use because you don't have a future application. So the first thing we do is different from many others. We say, okay, go to the clinical relevance and do that. And if you don't find any correlation with your patient tumor, forget about that. We move on. But this case, we are very lucky. And we find that in benign tissue, you can see the expression level is extremely low, if any, is located on, cyto or, cell membrane. And in cancer and in metastatic cancer even, so the expression is extremely high. And the location, the cellular locations are very interesting because they became localized in cytoplasm rather than only on the membrane. So a lot of things are interesting. And then we went on to trying to understand if the expression is higher in cancer, what is the mechanism over there? And there are many potential mechanisms. They could cause by genetic alteration, could cause by epigenetic alteration, so on, so forth. We're trying the fish analysis, mainly find whether or not it got a gene copy number change or amplification in prostate cancer. The answer actually is yes. So in normal situation, you don't have such amplification and copy number gain, but in cancer you do. So we partially understand why this gene and produce more protein. Of course, another question that you want to ask whether or not this is functionally important. If functionally important, that can be used as a target for therapy. And whether or not you can find a biomarker, that's a different argument. And that needs a lot of work. But we went on, we say, functionally, how you're going to test that. I don't have to go to the detail, just like everybody else, you do the in vitro functional test, you do the in vivo functional test, and then you come up with your conclusion. So this ASP1 seems working pretty nicely. And so I just want to give you a little bit of flavor. Actually, I have a big talk talking about a personalized cancer therapy, how to use such model for this. I don't think I have enough time to talking about that topic today, but I guess I gave you enough flavor to say you got comprehensive collection. And the method should be okay for even bladder cancer, renal cancer later on, like Peter and Alan are going to work with me together to expand this model to other urological kind of cancer. And so that should be all right, and I have confidence with that. And the bottom line, the application aspect, you can think about anything almost in the research field. So the model really have unlimited kind of useness, if you will. But you need to have an open mind for the model. Whatever model you pick up, you have to be, really objective and why you pick the model. And if you're too selective, that will give you a selective corner anyway. So I believe the model should be kind of more predictive, and this kind of model should facilitate the translational research all the way from discovery to the clinical development. This is really fit in with the Prostate Center's mission really well. That's why I'm extremely to be part of the Prostate Center and part of urology. And, I'm looking forward to more kind of collaboration. And this is the kind of argument I say, no, even your model is good, but remember, any model is only one corner of things and real things. And you have to be, realistic. And in order to have a good view, you need to use a panel model, and don't just try to say my model is perfect. No such things in the world. And what I'm trying to say, and no matter you call this one living tumor center or living tumor unit, and when everybody in the dark, and you hope this kind of strategy is going to give you a light. I think that's what I'm trying to say, and the living tumor is a solution for many things. So this work, I'm extremely lucky, and a lot of people working and have a faith on me and support the work, and particular my former mentor, Jerry Cunha. He's a great scientist. He probably regarded as the father of epithelial stroma interaction in the world, and then he published probably more than 400 publications. This kind of gentleman is really truly, friends who give me all the support from the very beginning, which I don't trust myself. He trusts me. He said, YZ, trust me, you can do, if you touch anything, you can do much better than most of the people. And I think that's really, I owe him a great deal. And a lot of people in the lab and over the years. Another good news is my lab from day one, and every day they came growing, and no single people actually left. And that's became my burden actually, and I need Larry's support to maintain my lab.